And yet, selection never acts on one locus on its own. It acts on individuals, and there's loci are. So what I want to do today is to expand our understanding and the kind of mathematical underpinnings of how selection works when there's more than one gene under selection. And you've already, this has come up again and again and again, the idea that there's hitchhiking and there's effects of selection on nearby sites. So today I just want to give you the mathematics behind that. But just to orient us, this is an example of a very common ornamental flower in North America, Primula vulgaris. And it has two floral forms. It has the female parts, the pin, and then the thrum, the male parts. And Darwin wrote about, you tried this out and it worked. Okay, yes, I have to say it, got it. Okay. Darwin published the first description of the pin and thrum mating system, and he wrote, in which the pollen of A is fitted for the stigma of B and conversely. You can see that in his drawing here. So that this is where the pollen comes from. This is where the pollen lands and fertilizes the seeds. And you can see this fitting that he talked about. It's called heterostyle in Primula. And it's one of the classic plant breeding system polymorphisms that we know about today. We've now, the genes underlying this polymorphism are now known, and it turns out that there's two loci underlying it, not one. So tall versus short female parts are underlined by alleles at the G locus, and the male tall versus short parts at the A locus. So how are these two morphs maintained? And why don't we see everything in between? Why don't we have the short male and female parts? To understand that, we have to understand the dynamics and selection acting on two genes, the A and G. And this particular case, there are some recombinants, but by and large, these are the genotypes that we find in the population, where A and capital G give the thrum morph, little a and little g the pin morph when homozygous. And that's just one example of bazillions of examples I could give you of how genes are embedded within chromosomes and interact with one another. So far, we've kind of had what has been called a beanbag genetics approach, looking at a gene as if it were isolated. It's never isolated. A site under selection is embedded in a gene, embedded in a structured chromosome, and that's embedded in a genome with other chromosomes. So how does that embedding affect in evolution? So over the last, I think, 30 years, we've really developed a multi-locus framework for understanding selection at genomes, not just at sites. And I'm going to touch upon some of that work. So just to orient us, I'm going to talk about locus. That's the same root as location. It's the site under selection. So I'm going to talk about multi-locus models, multiple sites under selection. And to build up the math, I'm going to do this focused on two loci, A and B, with two alleles each. And that's the simplest multi-locus model we can consider. And if they were evolving independently, then the beanbag models would work. And knowing which allele an individual carries at one locus, say that individual carries big A, would provide us no information about anything else in the genome. That's what we mean by independent, that there's no information provided by knowing what an individual is here about anywhere else. Just some more notation. We're going to track the allele frequency, but now at two loci, so big A at this locus and big B at that locus. Okay, so if everything were independent, this is putting that independent assumption into math. So these are just chromosomes, and I'm just counting up the chromosomes in a population that carry these four configurations. And if they were independent, then the frequency of this type of chromosome would just be the product of their component alleles. So if I have big A at the first site, then there's no information about whether or not it carries big B, and its frequency of big B is just the frequency of big B in the entire population. That's the independent case. So I'm going to use X's to denote the frequency of these genotypes, and the X's would sum to 1, and they sum to 1 in this particular example. But this is the assumption that we make that if the sites were independent, acting independently, evolving independently. And so if that were true, and there's no additional information about the other locus, if we look at one locus, then we could model it just the way we've been modeling and ignore associations. But what if there are associations? In the general case, there will be associations, and we'll talk about why they develop and how they're maintained. If it's not the case that the chromosome frequencies are the product of the component allele frequencies, then you can't just look at one locus and know in isolation and know how it will evolve. Because if the other locus is also under selection, their interaction will matter. So in order to build up models with 2 loci or more, then there's 2 major tools we need in a toolbox that we haven't yet put into our population genetics models. And one of them is linkage disequilibrium. It's come up a lot already. It's an association measure, and the other is recombination, which is a core process that you've learned about in genetics, but I'm just going to refresh your memory. So let's start with disequilibrium. Disequilibrium between 2 loci is also known as linkage disequilibrium or gametic phase disequilibrium, because it's about the alleles carried in the gamete. Some people use LD, but I'm going to use D for that association when we're talking about gametes sampled from within a population. There's other genetic associations. So remember when I talked about if heterozygotes were really fit and the homozygotes died, then there was an association between big A and little a. That was within a locus. That was a departure from Hardy-Weinberg, but that's still a genetic association. If you have homozygotes at one locus, if you have selfing homozygotes at one locus, you're more likely to be homozygous at another locus. That's identity disequilibrium. It's another type of genetic association. So I just want to park that. There's many types of genetic associations depending on what you're looking at. We're just going to look at neighboring sites. So the disequilibrium is defined by the product of this chromosome and its opposite chromosome in frequency minus the 2 that I've drawn in the middle here. So that's its definition. There's another definition, and I think Mike mentioned this, and that is that the disequilibrium measures the departure from our expectation of the frequency of a gamete and what its frequency is expected if they were not associated, which is the component allele frequencies. So if the disequilibrium is positive, we're going to see more of this chromosome than we expect, less of these 2, and more of that one. And if you sum those 4 up, it still sums to 1, so everything is good. The disequilibriums cancel when we sum this up, and the PA times PBs turn into a product that equals 1. Okay, so if there is disequilibrium between the 2 loci, then knowing that this individual carries big A provides information, says that if it's positive disequilibrium, it's more likely to be carrying big B than anybody else. Let's take a look at some examples. So I've concocted this example, it was just out of a hat. And if we multiply this frequency times that frequency, oh, thank you. And if we can we get rid of, there is a way to get rid of that thing at the bottom. What is it? Hide video panel, meeting control. Yes, all right. So if we multiply this frequency times that frequency, subtract off that product, we get a positive disequilibrium in this case. And that's saying that, These two, a positive disequilibrium means these two are chromosome combinations are more common than we'd expect. These two are less common than we expect. And so if we are carrying big A and there's positive disequilibrium, we're more likely to be seeing a gamete that also carries big B than we'd expect by chance. Same with little a. If we carry little a, then we look at the other locus, we're more likely to be carrying little b. And that's what we have with primula. We have very, very strong positive disequilibrium, where if you're carrying big A, you're almost certainly carrying big G. Of course, there's the opposite configuration, that these two chromosomes may be more common. In this example, we've got a negative disequilibrium. And so if you're carrying big A and you look down the chromosome at the other locus, you're more likely to be carrying little b. Okay. So just to sum up, linkage disequilibrium is a measure of genetic associations indicating which alleles tend to be found together at two loci. As Mike mentioned, it's terribly named, and as we'll see, you can have linkage disequilibrium even if they're unlinked, and you can have disequilibrium at equilibrium. Okay, the second tool we need is a measure of recombination between those loci, and that is going to be described by this recombination rate, little r. Recombination occurs during meiosis, and when chromosomes come together during meiosis, there's a possibility that that double helix is broken and recombines with a homolog chromosome. And that allows new combinations of alleles to be passed on to the offspring. So let's walk through this meiosis. If you have a diploid parent of this configuration, then right before meiosis, the chromosomes double. And then these, I've listed all of the possible gametes that can be formed from that diploid parent. Let's focus on the top and the bottom. Those are the parental combinations. If there weren't recombination, that's all we'd have, or the top and the bottom without recombination. And as long as meiosis is fair and there's not meiotic drive, then half of the parental chromosomes will be this one, and half of the parental chromosomes will be that one. The other two chromosomes are the ones produced by recombination. So with a total rate of R, we're going to get a recombinant chromosome. Half of them will be this recombinant, big A with little b, and the other half this recombinant, little a, big B. And those all sum to one. So if you're looking at a gamete, you've got to have some type of gamete. The type you have is either parental or non-parental. It's non-parental with probability 1 minus R. Non-parental with probability R. Parental with probability 1 minus R, and split into the two halves. One other point that I'll mention is I didn't choose this genotype randomly. I chose it so that it was different at the two loci. If this was big A, and that was the same, then it doesn't matter whether I recombine or not recombine. I'd always have half the gametes would be big A, big B, and the other half of the gametes would be big A, little b. Recombination wouldn't matter. It's still happening, but it doesn't mix up anything if you don't have differences at both loci. So recombination only enters into the propagation of gametes when those parental alleles differ at both loci. Okay. So just to recap, linkage disequilibrium is a measure of association. I think somewhere in my slides I didn't mention it's a covariance measure. So it's saying, do the alleles that I carry co-vary with the alleles at another locus? Whereas R is a measure of the genetic distance and how likely it is that there's a double strand break during meiosis that causes one chromosome to be broken apart and merged with the other one. Okay. You go into a population, you sample the population of squares and circles at two loci. What can I measure from this? How many people think you can measure from this snapshot of a population the linkage disequilibrium? How many people think that you can measure the recombination rate? Okay. You cannot measure the recombination rate because you don't have the parents and you don't know whether they had recombinant or non-recombinant offspring. So that you can't measure. You're not tracking meiosis. What you can measure is the linkage disequilibrium, because we can ask, is it more likely that you're going to carry a circle, a black circle if you carry a black square or not? But it's not obvious, and you have to actually do the calculations. So I went ahead and I summed everything up, and I said, at these two, the square locus and the circle locus, this is the number of chromosomes we have in our population. So go ahead and calculate the linkage disequilibrium. And when you have your answer, go ahead and raise your hand and lower it. And there's a little hint here that these X's are chromosome frequencies, so you have to first go from numbers to frequencies. I'll give it another couple of minutes, but for those of you who are done, think about what the biggest disequilibrium could be. What's the maximum that it could be? Okay. Does somebody want to volunteer their answer? 0.05. That's right. Oh, 0.05, let's move slides. So 0.05, and because that's a positive number, then that's telling us that these combinations are more common than we expect. And these combinations are more common or less common. No, it's not just one of them that's more common. If there's a positive disequilibrium, it's saying they're associated, and both of those are more common than the component chromosome frequencies predict. And it's not obvious. I can't just look at that. I have to do the calculations to figure it out. Now, some of you might be wondering, well, how do you decide what's big A and what's little a? And it's arbitrary. I've told you how to do it here. If you did it the opposite way, you would have gotten minus 0.05. But that minus 0.05, if you'd labeled it differently, would also be telling you that these very same things were more common. So you'll get the same associations no matter how you define your alleles. Okay. And how about the maximum? Did anybody figure out what the maximum disequilibrium would be? What was that? 1.1/4, exactly, 1/4, 0.25. And that's if these ones are equally frequent, a half, and those ones are absent. The most negative disequilibrium could be if that one's a half and that one's a half and these two are absent. But that's only possible if the allele frequencies are at half. If they're lower, you'll always get a smaller disequilibrium. So it's worth remembering that disequilibrium depends on the allele frequencies, and it has that maximum. Okay. So why am I talking about linkage disequilibrium? It's because most of the evolutionary processes we've been talking about generate linkage disequilibrium. Drift generates linkage disequilibrium. As a matter of fact, this disequilibrium could have been due to drift if by chance the black and black or the white and white parents just happen to have more kids. So this could just be reflecting drift. The lucky chance of which chromosomes and which parents reproduced. Mutation can generate drift. And this is a major signature that we see in genomes. And here is a picture that I drew from this paper. Imagine that we just label every chromosome in a population by a color. And a new mutation arises, and it's unique. And it happens in it's going to happen in some genome, and by chance, it just happened in that red genome. And so that means when that mutation first happens, anything else that is carried that differentiates that chromosome from the other chromosomes is in linkage disequilibrium with that mutation when it first arises. So here's that mutation from a C to A here, and that A is going to be in linkage disequilibrium with whatever else is carried by that red chromosome, just by chance of where that mutation hit. Another process that generates linkage disequilibrium is migration. If we consider migration over space, and we have a genetically distinct population over here and a genetically distinct population over there, then when in places where we find migrants from both populations, they're more likely to carry the red alleles everywhere or the blue alleles everywhere on those chromosomes where they come together. And so we expect if we mapped linkage disequilibrium over space, it'd be low where one allele, one haplotype is common, low where the other haplotype is common. And in the hybrid zone, we'd expect to see predominantly big A associated with big B and little a associated with little b, mostly. Now, of course, those are going to recombine, and it's not going to be the only types of chromosomes that we'd see in the middle, but they're going to be overrepresented because of the gene flow from that parent and the gene flow from this parent. So this is one of the ways that people scan for and identify hybrid zones and where they are. All right, selection. So some chromosomes may confer higher fitness, and those combinations will then be expected to rise. So I want to now dive in and say, okay, how much disequilibrium do we expect selection to build, and under what circumstances? So I'm going to go back to a model of selection, and I'm going to keep us focused on the simplest case of haploid selection. But many of these models have been built for diploids as well. So we're going to focus on selection in the haploid phase and go through the life cycle again, sexual life cycle again. Okay, there's a lot of bookkeeping about to happen, but we'll get to a state where we can really draw out some very important principles from the model. And it's only bookkeeping. So we're going to start at the gamete stage and count all 4 gamete types up. Those are the 4 X's, and they sum to 1. Then we have random mating. And so imagine that we randomly combine an egg of the 4 types and a sperm of the 4 types. And there's random mating, and so the chance of seeing this chromosome and that chromosome come together are just the products of the 2. Okay, there's no selection in the haploid stage, so nothing happens there. But now we have to consider meiosis and the break up of these. These are now the diploid zygotes that are produced. No selection in the diploid phase, then they're going to undergo meiosis. But recombination is going to act during that meiosis, and that's what we have to account for next. Okay, so now we want, what are the baby haploids produced after meiosis? And we're going to start with a big A, big B chromosome. Okay, so we look in this, this combination of diploids, and we look at all of the possible individuals that could make a big A, big B haploid kid. This one can't, it doesn't have big A, it doesn't have big B. This one can't either, it doesn't have big A. So there's some that can't contribute to that genotype without mutation. But there are some that can. First of all, everybody from this diploid parent is going to be big A, big B. Then there's a whole bunch that have one locus the same and the other locus polymorphic, and they're going to give 50% frequency here of big A, big B, 50% frequency of big A, little b. But we also have, this is reflected, we also have one where the sperm and the egg were the opposite type, and it's the same, X big A, big B, X big A, little b. So there's really 2 of these, and half of them each contribute to that. So the 2 types times a half contribute this amount here. Same with this genotype where the second type is little a, big B, half of this will contribute, half of that will contribute, they sum to this term. And then we have the more complicated ones, the 2 combinations where they're called double heterozygotes, where both loci differ. And there we have to account for whether or not recombination happens. This parent can only produce the big A, big B offspring if there's no recombination. So it has this times 1 minus R, and there's a second one down there, each of which has a half a chance of making the big A, big B chromosome type. So 2 of these types, and this is a chance of producing the parental big A, big B chromosome. And then last, we have this final type that can produce the big A, big B genotype, but only if there's recombination. because it doesn't have it big A, big B in either parental type. But if there's recombination, and we have two of them, and each of them has an R/2 chance of making this gamete. Okay, so let's do some simplification of that. I'm going to draw together all of the terms that involve recombination. And when I do that, I get this double heterozygote and that double heterozygote. So those are that term and that term. And then I'm going to factor out a big A, big B from all of the other terms that don't involve recombination. But when I do that, you can see it's X big A, big B times the sum of all of the gamete types. And that's just one. So I can simplify it down to X big A, big B minus RD. That's just one of the gamete types we can produce by recombination. But if you went through the same sort of logic, you get all of these other four types right after meiosis. And just as a check, they should sum to one, and they do. Those last terms will all cancel, and they'll sum to one because we had the gametes sum to one to begin with. Okay, now we're going to take them through selection. These are our four haploids. They witness selection given by these W's. And those would not sum to one, except that we divide by the mean fitness, which is the sum of those four types. And that ensures that they still sum to one, and it measures the mean fitness. So this is where everybody okay? It's just bookkeeping. But actually, these equations end up being pretty profound and helpful to understanding what's going to happen in evolution at two loci. So I want to derive the results and the implications of these equations. Oh, and just to go from this slide to the next slide, well, all I've done is replace these chromosome frequencies after meiosis with what we calculated them to be. And so that's how I got to this. And that takes us through one whole round of gamete union to produce diploids that undergo meiosis with recombination and selection. And that's the recursions. Okay, first point. If there's no selection acting at all, and W big A, B, all of the W's are one, they're all equally fit. W bar will be one, no fitness difference. Then what's going to happen over time? Well, let's track any one of the allele frequencies. Let's track the big A frequency in the next time step. So who carries the big A allele? Both this chromosome X big A big B and this chromosome X big A little b. Those are the only ones that do, so let's sum those two up. So I take the first two, these two terms, the fitnesses are gone. And when I do sum those two up, you'll notice that R and D disappear. So they're gone. And all I'm left with is X big A big B and X big A little b in the previous generation, which was the allele frequency in the previous generation. Give you a second to absorb that. Question? Yes. So we'll get to, maybe we'll get to that in the next slide. But notice that this does not depend on the recombination, and it doesn't depend on there being no linkage disequilibrium. This is saying the allele frequencies will stay constant from one generation to the next, regardless, if there's no fitness differences. So the breakup of recombination, of disequilibrium by recombination, just reassorts things, but it doesn't make one allele less common. So these are haploid though, so they're not at Hardy-Weinberg. But I think what you're saying is, what about the associations between big A and big B? So in the next slide, we talked about that. Oh, well, first implications. So considering multiple loci doesn't in and of itself cause allele frequencies to change over time, as long as fitnesses are equal. But now I think this is what you were going to get to. Just because the allele frequencies don't change doesn't mean the chromosome frequencies don't change. And indeed, they do change. And so if big B, A, and big B were more likely to be found together by chance, then in the next generation, that's going to go away a little bit because of recombination acting on that genetic association. So This isn't about Hardy-Weinberg because it's at two different loci. Hardy-Weinberg is about associations or not associations for the two alleles at a single diploid locus, but it's still the case that we can talk about genetic associations here between the two loci. If there's no disequilibrium, then this won't matter. If they're not genetically associated anyway, then it doesn't matter if there's recombination or not. Recombination only acts and only matters to evolution to the extent that there were associations there to begin with. So evolutionary change in allele frequencies is not expected due to recombination, but evolutionary change in chromosome types is expected because of the mixing that recombination brings, even without selection drift or mutation. And so in this example, if there's no further mutation or anything else going on, I said that when the mutation first appeared, it was associated with all the red alleles, whatever they're labeled, throughout the whole genome. But over time, recombination will act, and it will break apart all of those genetic associations and mix them up. And that's this process. And the amount of that mixing is at rate R. So we can calculate actually how the disequilibrium changes from one generation to the next using the equations that we've derived. So what we're going to do is we're going to take this, to get the disequilibrium in the next generation, we just do the same calculation. The product of those chromosomes minus the product of those chromosomes. But we're going to do it in the next generation, and we're going to plug these equations in. But there's no, again, no selection yet that we're considering. So this is the frequency of big B that we've calculated, little b that we've calculated, and this one. All good? So I'm going to then expand those terms and gather together everything that does not involve recombination or disequilibrium. So that's this term comes together. And everything else will involve recombination, and that term comes together. What's this? Disequilibrium at time t. So that's going to go to this term. Okay, and let's gather all the R terms. Minus R dt times x little ab, minus R dt times x big B, et cetera, et cetera. I'm gathering all of these terms together. Everything that involves the minus RD. What does this term in parentheses sum to? 1. It's just the sum of all the chromosome frequencies. So that all sums up to minus RD. And finally, there's a product of R squared D squared, but it's positive here, positive R squared D squared, minus positive R squared D squared. And that all cancels out. And that gives us that the disequilibrium from one generation to the next in the absence of selection decays by a factor 1 minus R. That's come up in lectures before. And so this is just showing you that decay over time with recombination of the initial disequilibrium. Here I started it at the maximum possible disequilibrium, 0.25, and just ran it. So a couple of key points. That even with unlinked loci, it doesn't go away immediately. Hardy-Weinberg is approached immediately in one generation with random mating. But having no linkage disequilibrium, no associations between the loci, that takes time. Why is any idea of why it might take time?
Disequilibrium comes because of uneven association.
Yeah.
Recombination rectifies it. It takes time for these things to come back.
Yeah, it does take time for them to come well mixed. The way I think about it is that not all chromosomes are going to be in double heterozygotes, right? Some of those chromosomes that bear these associated genes aren't affected by recombination. They're in homozygotes or double homozygotes. And so recombination can only untie these associations if they're both present in the same parent during meiosis, both big A and little a and big B and little b. So it can't make every association disappear immediately. Yeah. Yeah, so I've been, notice if there's no selection, this math is true for any sexual, because this is a sexual, yeah, there's no selection in the diploid phase, no selection in the haploid phase. We did a sexual cycle. So this doesn't depend on whether we're looking at haploids or diploids. Yep. Yes. That's right. That's right. And we've kind of, if we go back a bit, right. So this table here is the two locus equivalent of a Hardy-Weinberg table. We've assumed that the eggs and the sperm are randomly united. And so if we were focusing on one locus, we would find Hardy-Weinberg at that one locus. But if we looked at, if you were homozygous at the big A locus and there was disequilibrium, you'd also be more likely to be homozygous at the big B through this linkage disequilibrium. Okay. So even unlinked loci R equals a half remain associated for a while. And that's because not all chromosomes are in double heterozygotes to be broken apart by recombination. And the other thing about this result is that it means that these genetic associations will persist for longest if at nearby sites. And that underlies the type of tests that Mike was talking about, these GWAS or other tests, because you look to see whether or not there's associations between a particular trait of interest and markers. So this is data from the human chromosome demonstrating that linkage disequilibrium is indeed largest for sites that are close together, and it dissipates with the distance between them. Here, linkage disequilibrium is measured as the correlation measure, R squared. Okay. And this has been, the fact that you expect nearby sites to stay in linkage disequilibrium for longer has been used to find the genetic basis of diseases. The first of these studies was by Mary Claire King, who was looking for the cause of familial breast cancer that ran in families. She had pedigrees from 1,500 different families of when did the daughter have the cancer that the mother had or the grandmother had. She tracked 183 markers at the time. That was a lot of markers across the genome. Now it would be a teeny one. And at the time she was told not to do it. She's like, this is gonna be a needle in a haystack. You only have 183 markers across the whole genome. What are the chances that you'll land near enough to the genes causing cancer to even find them? But she was stubborn, and stubbornness is a good trait in a scientist. And she's like, I'm gonna do it anyway, see if I can find something. So she tracked these breast cancer and ovarian cancers through these families. And that led to the discovery of BRCA1, one of the most important genes contributing to heritable breast cancer. So what are those markers? I think you probably are familiar, but those are polymorphic sites that can be traced among families. So she was looking, what genotype do those have at all of those markers? And that's what she tracked. And typically, you know, she had 183, she was not actually finding the markers weren't the ones that were responsible for the breast cancer. She was just hoping they were in tight enough linkage disequilibrium with the sites under selection, the sites that were causing the breast cancer. And so, and it continues to this day. We can find what sites are responsible for local adaptation, like Mike talked about, or from disease by looking for signals of nearby sites that are perturbed or changed by the trait of interest. Okay. So far I've been talking about without selection, but let's add selection to this. Now let's say only one of these loci is selected and not the other one. Yeah, question. Right. Well, so if there was no disequilibrium, if you made an infinite population with none of those other processes and there was no disequilibrium to start with, it would stay at 0. if you had some disequilibrium to start with, and it was the only process acting, and every generation it decayed by a factor 1 minus r, then you're right. It would take kind of infinitely long to go away, although it would decay exponentially. So after a while, you wouldn't notice it. Yeah, but that's right. It certainly doesn't go away right away. And I think in one of the previous lectures, there was an approximation made that was this. And that's just, this is the exact result for a discrete generation model. But if R is small and you're talking about linked sites, e to the minus r is approximately 1 minus r. All right, okay, let's derive this. Where I want to get to is I want to say A is under selection, B is not under selection. What I want to show to you is that the dynamics of A are the same as what they would have been before. So the neutral locus doesn't affect the selected locus. But I want to show you that the selected locus does change what we see at the neutral locus. Okay, that's the goal. And we can get that up here. What was the name of this process? It's come up before, where selection at one site influences what we see in another. Sweep, yeah, it's genetic sweep. So selection at this site sweeps away heterozygosity at neighboring sites. So yeah, that's a good answer. Hitchhiking was the other term. Yeah, and I think I heard it too. Okay, so we want to see how much hitchhiking do we expect as a result of selection. All right, so I need to calculate the change in the big A allele frequency. So to do that, let's start with just the big A allele frequency in the next time step. And that comes from two genotypes, X big A big B at time t plus 1, and X big A little b at time t plus 1. So this is just counting up who carries big A. And then from my equations, now we have selection included, so I'm going to put it in. I'm going to drop the T notation so I don't have to write T all the time, okay? And I have W big A big B times X big A big B minus RD over W bar plus X W big A little b times X big A little b minus RD over W bar plus RD. You guys have to keep me honest about my typos. Okay. But I'm saying that there's only selection acting on the A locus. So the B locus doesn't determine fitness. So I'm going to erase the B locus because it doesn't affect fitness, and we just have that. All right, so let's pull together the terms that don't involve recombination. It's always good to, when factoring, decide on a term that you're going to gather all the terms that involve it. So I'm going to pull the recombination terms out. And I've got a big X big A big B here. And I have a X big A little b there. And then we have to deal with the recombination terms minus RD times W big A W bar from the first one. And then we have a plus RD W big A over W bar from here. What can I, yes. Yeah. 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 Individual hap. Yeah. But now I'm saying B is neutral. I'll write that up. We only have selection and fitness differences that depend on A. We'll put it back in a minute. That's why I erased it. Okay, and so cleaning this up, that cancels with that. What's this? BA, thank you, I just didn't hear. So that's W big A, PA. I'll be explicit about time. And then W bar, if I calculated it, it would be, W bar is the sum of all of these numerators, but they don't depend on the B. The fitness doesn't depend on B. We get the same cancellation of the R's and the D's, and that just leads to W big A. I'm not going to do the derivation, but you can do it on your own. Just, and that's what we had before. This is selection on the A locus, depending on the fitnesses of the A locus. No influence of a neutral locus, even if they're in linkage disequilibrium. Are you is that comfortable? Is it pretty much what you'd expected? Yeah. So it's, it's like in physics, I guess, if there's not a force pushing it, then it will stay where it was, and there's no force pushing A away from its dynamics. Okay. And this is just calculating the change in A if you want it from one generation to the next. But I think that's enough. Okay, now let's look at the other locus, though. How much does B change due to selection at A, even though B is neutral? Okay, so I've got to, now I need the big B's. So the frequency of big B is, I sum up all individuals that carry big B, and then I plug in the recursion equations for that. And that is, and then the other one that carries big B is, but now again, B doesn't affect fitness. So I'm going to erase that. And now I can't factor out a W big A because there's different W big A's in there, but let's just start the process of factoring out. I'm going to move the recombination terms over to one side, and I have W big A, X big A big B, plus W little a, X little a big B, over W bar. And then I have recombination terms. Minus RD W big A over W bar. And then plus RD W little a over W bar. Okay. A long, long time ago in my slides, a long time ago, when I said, what does this equilibrium measure? It measures associations between the loci. And I should have written it on the board, but I pointed out there was another way to write big X, big A, big B as the frequency of its component alleles plus the disequilibrium. This is just another way of writing these genotype frequencies. And I'm going to do that now with these two chromosome types. So just to recap, you can count chromosomes based on the frequency of each chromosome, or you can count chromosomes by the frequency of the alleles and the genetic association between them, and both are equivalent mathematical descriptions of the system. This only involves 3 variables because the x's sum to 1. So this only involves 3 variables effectively too, for those of you who are wondering about numbers of variables. All right, so we're going to replace this with P big B, big A, big B plus D. We're going to replace this with P little a, P big B minus D. Okay, let's clean that up. So we have a W big A, P B, plus a W little a, P A B over W bar. That's ignoring the D terms. Then I'm going to have the D terms. I have plus W big A D minus W little a D over W bar. And then I have minus RD. I'm going to put these two terms together, W big A minus W little a over W bar. Okay. This first term, I can factor out a P big B. And then do you recognize this top thing without the PB? Once I factored out that PB, what? The mean, what did you say? It's the mean, we just saw it on the denominator before. It's what? It's the mean fitness, it's W bar. In this case where only the A locus is under selection, then the mean fitness is how often are you big A and what's your fitness? How often are you little a and what's your fitness? So those W bars just cancel and we get PB out of this first term. I want to factor this. I'm going to take a disequilibrium out. And I get that. And then I can kind of see, oh, the fractions are the same in the two cases. So what do I have left? So I have overall, in the next generation, at the B locus, I have the frequency at the B locus in the previous generation, plus I have the disequilibrium times W big A minus W little a over the mean fitness times 1 minus R. Okay. So this is a neutral locus. When will the neutral locus change from one generation to the next? If the fitnesses are different. Yeah, so if this is non-zero, then you're going to see frequency. But this is fitness at the other locus. If there's selection at another locus, that's required. What else is required? R can't be 1. But remember, R is the fraction of non-parental chromosomes, and so the maximum it is a half. So what else do we need? D can't be 0. So whenever there's whatever association between loci for whatever reason, we're going to see a change from one generation to the next at a neutral locus, even though it wasn't under selection. So we have to be careful because it can look like it's under selection when it's not. Okay. And this was just subtracting, the change in B is just the difference. Subtracting PB at time t from both sides, and that gives us this term here. All right. So we've now derived how much genetic hitchhiking we expect to see at a nearby locus. So selection at one site causes changes at other sites that are in linkage disequilibrium with it. Typically, the amount of change that you're going to see is bigger for nearby sites because that R is smaller, and the disequilibrium can persist for longer. One particular example is when a mutation occurs on one of the chromosomes within the population, only one. That beneficial allele happens on some chromosome with something, and as it rises in frequency, the nearby sites will rise in frequency along with it. And so you see that in this plot where the rise in the gold beneficial allele pulls nearby sites. It doesn't fix everything in this example because what had to have happened sometime between this locus and that locus? Recombination would have had to bring that allele, which was over here, into the same chromosome as the beneficial allele. So the more distant that site is, the more likely it is. So that's genetic hitchhiking. The process that selection acting on one locus will perturb the things that are nearby. And it'll perturb things across the genome, but if R equals a half, the disequilibrium goes away really quickly. And the stronger selection is, the faster a selected allele rises in frequency and the more sites hitchhike along it before being separated by recombination, which is really an important signature for finding selection in the first place these days. If this wasn't true, it'd be very hard. It would be a needle in a haystack because we'd have to be able to see a signature at that one site. And if it's fixed, we wouldn't know why. So we're always looking for things that are happening nearby. It's also the case that if big A rises in frequency, it can actually drive the deleterious mutations to rise in frequency as well through this hitchhiking. And that Matthew Hartfield and I analyzed what's the probability that a deleterious mutation would fix because of this hitchhiking. And that only really happens if the deleterious effect is milder than the strength of the beneficial effect. But it's much different than what Kavita showed you, what the fixation probability of a deleterious mutation would be. And it's negligible for large populations. But hitchhiking can cause the fixation of deleterious mutations. And what's interesting about that is that you might not even know. Two sites might fix. One of them might have been the beneficial one. One of them might have been the deleterious allele that got dragged up in frequency by accident, by genetic hitchhiking. And you may identify the wrong one as the one that was under, what's the cause of selection. All right. And now the last case that I want to consider is what if both loci are under selection? So now we want to deal with the full dynamics. So, and I'm going to simplify it just to the following question. If there's no associations between the loci to begin with, what would build up associations? That's what I want to answer with this set of equations. And that will kind of give you an idea of what what's required for independent loci to stay independent in their evolutionary dynamics for associations not to build. You ready? Okay. So what I want to know is if now we're not assuming B is neutral, we're assuming that we have no associations to begin with. But selection is acting at two loci. What will happen? So I want to know what's the disequilibrium in the next generation. And that will just be, by definition, the product of the big A, big B, and the little b chromosomes in the next generation, minus the intermediate types, whatever they are. It's bookkeeping, and there's a lot of that. I'm sorry, but we'll get there. Okay. So now I'm going to replace those with our equations, including fitness at both loci, but I don't have any disequilibrium. So I'm dropping by assumption. So I'm dropping that. And these are all at time t. So I'm going back a generation. All good? I didn't make any typos. Okay, so just to recap, I dropped all of the D's, assumed there's no associations, that everything's well mixed, dropped the disequilibrium, and now I'm calculating the disequilibrium in the next generation. And I'm going to use this same definitions to rewrite the chromosome frequencies in the previous generation into this other variable system, the alleles frequencies in the disequilibrium. There's a W squared, mean fitness squared under everything, so let's just stick that down there. So I have W big A big B. This is P big A P big B. It would be plus disequilibrium, but there was no disequilibrium in the previous generation. So it's just each chromosome is the expected frequency based on the component allele frequencies, because there's no disease. So I just marched through this equation. Okay. Does something factor out? The product of all of the allele frequencies drops out. They're in a different order in this one and that one, but they're all present. And that leaves us with just one term. So when will we have disequilibrium in the next generation if there wasn't disequilibrium before? What does it require? Yeah, very nice. It requires an association in the fitnesses. And that association in the fitnesses where being big A and big B make you more fit, or the other one, little a, little b, makes you more fit. If this combination, those two genotypes have on average a higher fitness than you'd expect based on the fitnesses of the other type, we call that kind of an association in the fitnesses epistasis. So you need epistasis to be present. And what else do you need? You need polymorphism you need all four of those alleles to be present in the population. Did I write them all? Yeah, I got them all. So you have to have polymorphism at both loci, and those polymorphisms have to affect fitness in a way that these are not equal to one another. I'm gonna, let's write that in another way. I'm gonna first use my hands. So imagine that your fitness surface looks like this. Every capital letter, every increases fitness. So let's just say this is, you have 0 capital letters, 1 capital letters, 2 capital letters. And if fitness is really strongly positively curved, then you're gonna be really fit if you have 2 alleles and on average, 0 capital alleles. And those have looked to have higher fitness. Whereas the ones in the middle, the ones with only 1 capital letter have low fitness. That would be positive epistasis. Or it could be the other way around. Maybe it goes like this. That would be positive epistasis. That would be negative epistasis. But it's worth saying, when would this be 0? What would have to be true about the fitnesses in order for them to be 0? And it might be obvious to you, but just in case it's not, it's when the fitnesses multiply together at the 2 loci. That'll be 0 if W big A big B is just equal to W big A times W big B. And if there were multiplicative interactions between the loci, multiplicative fitnesses, then we would just have that the fitness that you have as a big A, big B genotype is just the product of your fitnesses at each locus considered separately. Repeating that all the way through. This cancels with that. So in the multiplicative case, where fitnesses just multiply together at the 2 loci, that's kind of the null case, where we don't expect associations to build between the loci if they're absent to begin with. And you may have wondered in the literature, why are we measuring epistasis relative to multiplicative fitness? It's for that reason. There's no inherent reason otherwise. Questions? Yeah. I mean statistical associations. If you have no statistical associations, so it's like a well-shuffled deck of cards, and there's no associations between which card you see in the first locus and the second locus. If there's no associations, and you want to know what builds associations, you require that this not be zero. If the fitnesses are multiplicative, then if it's well shuffled to begin with, it'll be well shuffled in the next generation too. No associations between what you see at one locus and what you see at the other locus. Now the allele frequencies are rising because of selection, but you won't get associations between, correlations between whether you carry big B and whether you carry big A. Yeah, so functional associations would be saying you don't have epistasis. So for example, if big A, if this is a dimer and a protein dimer, the two loci are, and you have to have the alleles work well together, then this might be really high and everything else might be really low. And you won't have multiplicative interactions because of that fitness interaction. And you will have epistasis. And it will build up positive associations. If this one is astronomically high, let's say, you know, only the big A, big B genotype has really any fitness. Everything else doesn't have the right dimer formation. It's not working. Then this one's much higher than those. This will be a positive epistasis, and it'll generate a positive disequilibrium. So you'll see more chromosomes that work well because they carry the two than you'd expect because of past selection. So past selection will have favored those that had higher fitness, those gene combinations, and they come to be at a higher frequency. Yeah. The loci have fitnesses that are multiplicative. What's the opposite? Yeah. So the opposite is that there's epistasis. So if it's multiplicative, we're saying there's no epistasis on this scale. And if W big A big B, W little a little b is, if the extremes are more fit than the intermediate ones here, I'm labeling the alleles so that we're talking about some trait of interest where every capital letter is increasing your trait or whatever. So if the extremes are on average, geometric average of the extremes is higher than the geometric average of the intermediate ones, then we say there's positive epistasis. If it's equal, we say there's no epistasis. And then if it's less than, we say there's negative epistasis. Sometimes in the literature, those are called synergistic and antagonistic epistasis. I don't like those terms personally because this is synergistic epistasis if big A, if little a and little b are beneficial alleles, but it's antagonistic epistasis if they're deleterious. So it requires that you flip how you think about it. But just talking about positive and negative, it's really talking about the curvature of the fitness surface. Is it more curved than you'd expect with multiplicative interactions? Then there's positive epistasis. Less curved, then it's negative epistasis. And whatever the curvature is, if we have no disequilibrium to start with, then this will create positive disequilibrium. This will create negative disequilibrium, and that won't create any disequilibrium. Things get more complicated because you can have other sources of disequilibrium. So you could have negative disequilibrium despite the fact that there's positive epistasis. And then there's some transients and things before it settles down. To zero? Yes, we have just proven that the only if you have variation at both loci and no associations to begin with, if and only if this numerator is non-zero, will you have disequilibrium in the next generation? I see what you're saying. Yeah, I think if there's no other no other interaction that will cause it. If yes, you just need this product to be greater than this product, but for that product to be greater than this product, it is stating that the combinations of alleles are working better together than you'd expect. Yeah, but you're right. I haven't formally proven that those are equivalent statements. I'll see if I get that for you. But this is how we measure epistasis. All right, so back to this example. There's two main reasons why the pin and the thrum morph are all that we see, and we don't see the recombinants. One is because the matings that work well are the ones where the pollen matches up to this style, or to the top of the stigma, and so there's strong fitness benefits for the pin morph and the thrum morph. Those genotypes have high fitness. The intermediate ones that have short of everything or long of everything have lower fitness. So it has really strong negative epistasis selecting against those. The other reason is they're very close together in the genome, and so there's very little recombination, which also maintains this linkage disequilibrium. Right. Another point. So if both sites are under selection, but there's recombination as well, turns out that the proof that we were able to make for the haploid and diploid one locus model no longer holds. Recombination with selection can lead mean fitness to go down from one generation to the next. And here's just a sample example simulation, starting with these chromosome frequencies where you get mean fitness going down and then going up, even if R equals a half. And one way to think about this is that if you have the fittest genotypes happen to be at really high frequency, recombination can break those down and create a lower offspring fitness than you would have had in the parents. So there can be more breaking apart of good gene combinations than selection is building them up. So evolutionary change in a genome will not always increase adaptation, and evolution doesn't always optimize populations. Instead, it works through these combined processes of selection, mutation, recombination, and drift. There is this strong hill climbing aspect that adaptation at any one locus is tending to increase fitness, but other things like recombination can undo that. Well done. And you can even, not in the haploid case that I know of, I'm pretty sure, but in the diploid case, you can even have persistent cycles, even if all of those fitnesses are constant. So this is a figure from Hastings in 1981, Alan Hastings, where he showed using bifurcation theory that if you there's a range of fitnesses that allow cycles to persist over evolutionary time. And it involves kind of some under dominance. So you are first at this homozygote rises, but then that favors this heterozygote at the other locus, which then drives down the homozygote. So it creates these persistent cycles. And it's not clear to him, and it's not clear to me how often these kind of fitnesses arise, but it's not impossible. And we would be misled into thinking the environment was causing it, these fluctuations. Or if we only had one snapshot in time, we might go, oh, there's directional selection, when it's just about to reverse. So, right, at this locus, diversity goes down and then up and then down and then up. And you might want to ask, well, what about these oscillations? How do they influence the neutral sites outside of this region. And yeah, those oscillations will also kind of reduce the effective population size at neighboring sites outside of these two. But yeah, half the time diversity is going up and half the time diversity is going down at those loci. And you can also, remember I said linkage disequilibrium is badly named, and you can maintain linkage disequilibrium. So there's some kind of classic models to try and find out what is the equilibrium in these models and under what conditions would we see disequilibrium maintained. So this is from a 1981 paper from, again, Alan Hastings. Yeah, I think that's right. And what I'm showing you here is the recombination rate between the two loci, and the dark bolded line is the amount of disequilibrium between the two loci at equilibrium. And this is with multiplicative fitnesses. So let's park that in your head. Let's talk about that in a second. But what he sees is that what Hastings showed is that with a tight enough recombination, you actually maintain really strong disequilibrium. So you tend to either see big A, big B, or little a, little b. Or you see the opposite. And if you work through the fitnesses, you could convince yourself that actually having these combinations or these combinations are more fit with multiplicative epistasis than the multiplicative fitnesses. And I should say that this had was diploid and had heterozygote advantage at the loci. No, they're not. So the fitnesses weren't symmetric at A and B. If they are, then you do get symmetric disequilibrium. In the fully symmetric model, you get exactly symmetric disequilibrium. But without symmetry in the fitnesses, as in this example, you can get two simultaneously stable equilibria, one where there's no genetic associations and one where there is. And where you go to just depends on where you start. The unstable equilibrium in between. Yeah. Now you might go, but Sally, you just promised me if there was no epistasis on a multiplicative scale, fitnesses were multiplicative, then disequilibrium wouldn't build if it wasn't there. And that's true. There is an equilibrium at D equals 0. If you start there, you stay there. But if you don't start there, you can move away from there and you can build up disequilibrium. And almost all models of fitness interactions tend to build up disequilibrium and associations and maintain them. If you have heterozygote advantage at both loci or something like it so that you can maintain polymorphism at equilibrium, you tend to see disequilibrium at least nearby. How relevant is this? Well, it depends on how strong selection is. This is a really small window, but that's partly because the selection was kind of weak. The stronger selection is, the bigger that window, the weaker selection is, the narrower that window in which you see associations maintained at equilibrium. And one little weirdness is that the one model that does not have any linkage disequilibrium at equilibrium has additive fitnesses across loci. That is W big A big B any of them is the average of the fitnesses expected at the two loci. This is called an additive fitness regime. And as we just proved, that doesn't obey, that doesn't obey the condition of no epistasis. So this epistasis isn't 0 if you calculate it at that fitness regime. So it won't stay at 0 if there's no associations, but eventually wherever it gets to, there won't be any disequilibrium. And I just mentioned that because you could say, well, maybe that's a more natural way to measure epistasis, but we don't. We measure it on the basis of those multiplicative interactions. All right, I think that's enough of a recap. Linkage disequilibrium is measured by this formula, and it tells us how much more common certain chromosomes are than expected. It can arise through all sorts of different processes. Without any other process that work besides recombination, it'll decay by a factor 1 minus R every generation. Linkage disequilibrium between a selected and a neutral locus can cause alleles at the neutral locus to change in frequency through genetic hitchhiking. And as I mentioned, that's also true for linked sites that are under much weaker selection. Deleterious mutations can hitchhike, and even weaker beneficial ones. With selection, linkage disequilibrium can be generated by fitness interactions. Those are called epistasis and can persist indefinitely at equilibrium. And finally, mean fitness can decline through the combined action of selection and recombination. And we can even get cycles in the recombination rate under those crazy conditions that Alan Hastings showed. All right. Questions?
So how much of these effects robustly drift? For example, these oscillations.
So they're robust as long as you maintain the polymorphism. It'll go back. This is a stable limit cycle that it goes to, not a neutral limit cycle. So it's stable as long as, you know, these are far enough away that you don't drift to fixation by accident.
Can go away if I'm sort of a selection.
Yeah, absolutely. I mean, he focused on pretty strong. So like these are very different fitnesses, but yeah, it's not clear. I don't know of any empirical case that has shown that it should cycle. Yeah, and if you work through it, you can kind of see. So what was this? This is lambda is really small, 0.03. So if you start in this corner, then this one's more fit. I'm not necessarily going to see which way to go. I mean, if you start here, then this one goes up. Anyway, I can't remember exactly which way it goes. But when you stare at this long enough, you can figure out where that cycle must be, but it's not. That one. What is this? So this analysis assumed multiplicative fitnesses, and this is an analysis of the disequilibrium with multiplicative fitnesses. I've worked on a similar analysis with symmetric fitnesses, and as I mentioned, Ewan's in this nice class, it's like a one-page paper, it's really nice, showed that with additive fitnesses at equilibrium, you wouldn't have disequilibrium. But it's not actually proven in general. These models are already complex enough with 3 independent variables and all of those fitnesses to know to prove what the disequilibrium is at equilibrium. That's right. So if you are, so if you have d equals 0, you'll stay at d equals 0. And that's an unstable equilibrium. So if by drift or anything happens and it goes up, then you'll move towards the positive disequilibrium equilibrium. Yeah. That's right. And even if you start way away, it'll approach it. Yeah. Yeah. So this is a bit sophisticated, but it's also, I feel like, a part of population genetics theory that we're kind of losing track of. These kind of classic, how do loci interact with one another? When do they maintain associations? When do they not? So I hope that this just gives you a little bit of a window into that literature. It was really a classic population genetics question in the 1970s and 80s. Thank you. That's it. I'll take more questions, so if you have any. Can you define D between different chromosomes? Yeah. And the definition that I used, X, like the chance that the kind of extreme chromosomes are found relative to the ones intermediate. However you define those alleles, those loci could be on different loci, different chromosomes. They could be on the opposite side of the same chromosome, but they could be on different chromosomes. If they're on different chromosomes, then recombination is a half. And so we would expect the disequilibrium to go down by a half every time step. And that's because the parental and the non-parental chromosomes are equal, and so R is a half. Yeah, so that you don't expect it to persist for very long. If you see disequilibrium persisting, it's a sign of which process of all of the ones we've talked about. Yeah, so selection could do it for sure. If you had epistasis between different loci on different chromosomes. But what's the process that's most likely to generate positive disequilibrium across the genome?
The bifurcation diagram was the continuous models or discrete?
I gave two references at the bottom, and one's discrete and one's continuous. So it's seen for both. I think you need haploid selection, though. Sorry, I think you need diploid selection because you need to maintain polymorphism. So you should have some heterozygote advantage or something like that. How do I share this? There we go. Oh, I think I left. Anyway, at the bottom, I will circulate. There were citations for both discrete and continuous time. And there was another question. Oh, yeah. So I said that drift, mutation, migration, and selection can all generate disequilibrium. Which of those is most likely to do so across the whole genome? Think about those frogs. Migration, because it's whatever genetic differences are between your populations that are coming together, you're going to see a overrepresentation of that parent species chromosome combinations, this parent species chromosome combinations in the hybrid zone. And so if you see disequilibrium across chromosomes, that's the most likely process, unless it's just one locus and then you think selection. Yeah. So that is a deterministic model, no drift. And what they do, remember we calculated equilibrium by saying, when will PA in the next generation be what it was in the previous generation? So now they're doing that for all four. Wouldn't X big A big B in the next generation exactly equal to X big A big B in the previous generation? And so if they have, so that model was saying if R is large, the only equilibrium it approaches, there's random associations, no linkage disequilibrium between the chromosomes. That's that flat line at R equals, at no disequilibrium, D equals 0. But if they're tightly enough linked, you go to an equilibrium either where big A, big B is overrepresented, little a, little b overrepresented, or the opposite, big A, little b is overrepresented. So, and then it stays. Even there's no drift, no mutation, nothing else going on. Whether you go up or down. Or you'll stay at 0 if you start at 0. But anywhere else, you'll move away from it. Mm-hmm. So in the lecture after lunch, I'm going to talk, this is a really good jumping off ground to going, okay, well, why do we even have sex in the first place?